Hi, I'm Miguel.
And I'm Heather.
We're both in the Department of Gene and Cell Medicine at UMass Chan Medical School, and we run the Translation Institute for Molecular Therapeutics.
So SPG4 is a neurodegenerative disorder that primarily affects big, long tracts in the spinal cord and the brain, and relentlessly causes these kids to lose the ability to have normal function in life. They lose their ability to walk, they develop slow speech, etc. And it never stops. It just continues their whole lifetime.
So we came together, a group of scientists and clinicians, to develop a gene therapy to replace the functional gene and to silence the bad copy that kids carry with them. We brought together multiple investigators from different institutions across the country and working collaboratively to develop a gene therapy to treat SPG4.
So what does this mean for Jack? So we have successfully brought 2 treatments to clinical trials for other neurologic diseases of kids that are fatal. And for Jack, what we're hoping to do is bring forth another clinical trial to treat him, to stop his disease progression and to help him live a normal life.
So our program starts with identifying what is the best gene therapy in animal models, and after that, we have to put together a program that goes to the FDA and asks if we're ready to move towards a clinical trial. Once we have the agreement from them, we do a toxicology study to demonstrate that the drug that we're going to bring to the clinic is safe. And then after that, we start to manufacture the clinical material for the trial. In the meantime, our clinician colleagues are developing a natural history study so that during the clinical trial, we can understand if the gene therapy is making a difference. And this is the entire program that requires a lot of scientists and clinicians working together to achieve success and help Jack.
Jack has a disease called hereditary spastic paraplegia. My name is Peter Voss, and I study Jack's disease at Drexel University. Jack has a variant of hereditary spastic paraplegia called SPG4. SPG4 is caused by a gene mutation that makes the nerves in Jack's spinal cord degenerate. This makes it harder for him to walk and run and do other things that are easy for most children. When I started studying SPG4 HSP, nobody knew how mutations of the SPG4 gene cause this disease. I've worked to understand this for quite a few years, and I've created tools that we can use to find treatments. In my lab, we now have mice with SPG4 mutations, and we've developed cell lines, antibodies, DNA constructs, and drugs to help us get there. I'm working on gene therapy, antibody therapy, drug therapy, and physical therapy, all of which will play a role in making life better for patients like Jack. It's probably not going to be any one thing, but a combination of therapies that ultimately stops and reverses the symptoms. SPG4 is most often adult onset. Childhood cases usually have the worst symptoms, but offer the most hope because a child's body is still developing. This means once we stop the mutant protein, there's a better chance of restoring what Jack's lost. This is a tough disease, but we won't give up. Jack gives me hope that we can get this done. Thank you.
Hi, my name is Leon Oscar Chang. I am an associate professor at Drexel University College of Medicine, and my research focuses on understanding and developing treatments for spastic paraplegia type 4, aka SPG4. SPG4 is a rare genetic disorder that causes axonal degeneration of upper motor neurons, leading to progressive muscle weakness in the lower limbs and difficulty walking. My lab is working hard to understand why and how SPG4 damages upper motor neurons by using SPG4 patient-derived motor cortical organoids and transgenic mouse models. Patient-derived motor cortical organoids are miniature lab-grown brain models generated from human skin cells to mimic the primary motor cortex, the brain region that controls voluntary movement. They provide a powerful tool for studying diseases like HSP and SPG4 and testing potential treatments in the human relevant system. These advanced models enable us to study SPG4 at the cellular level uncover relevant disease mechanisms, and testing potential therapies. Ultimately, it brings us closer to effective treatments of SPG4. SPG4 affected both children and adults, unfortunately, progressively limiting their mobility and independence. But I believe that through research, we can change the future for kids like Jack. Jack wants to be an astronaut and the first person to walk on Mars. Just like astronauts push the boundary of space exploration, we are pushing the boundaries of medical science to find a cure that will allow Jack and others to reach their full potential. This research is not just about science. It is about hope, mobility, and giving kids like Jack the freedom to chase their dreams. The work we do is made possible by your support. Every contribution to the SPG4 Cure Project brings us closer to treatments that could change lives. I would like to say, Jack, keep reaching for the stars. And to everyone supporting this cause, thank you so much for believing in the power of science and in a brighter future for kids with SPG4. Thank you very much.
Hello to the friends, family, and supporters of Jack. My name is Molly Letman, and I'm a researcher at the University of Wisconsin. My research focuses on hereditary spastic paraplegia arising from mutations in the gene SPG4. We developed a rat model of SPG4 that has been useful in testing potential therapeutics, as these rats recapitulate some key features present in patients with mutations in SPG4, including progressive decline in motor function, as well as changes to the connections between the brain and skeletal muscle that control voluntary movement. Jack's family, together with other families with kids with mutations in SPG4, have brought together several research groups studying SPG4 and have been instrumental in driving our research forward. Our typical method to fund our research is through grants from the U.S. National Institutes of Health. But this funding mechanism can be slow, as the preliminary data required for a research grant can take several years to compile, and funding is increasingly tight and unreliable. Charitable gifts, like yours tonight, provide us with the research funds to test new therapies without delay. Specifically, I deliver these to our rat model and measure how they impact disease progression. With your support, we hope to be able to help Jack and kids like him in the shortest time frame possible, which is really crucial, as we expect that any therapy would be best at stopping or slowing disease progression rather than reversing disease. My most sincere thanks to you all. Enjoy the rest of your evening.
Good evening, everybody. It's a great pleasure to be with you this evening. We're sorry that we can't be there in person. My name is Darius Ibrahimi-Fakari, and I'm a pediatric neurologist and neuroscientist here at Boston Children's. And with me today are Dr. Luca Schierbaum and Amy Tam from our research team. We have the pleasure of introducing our research program in the next 2 minutes. But before we go into this, I want to say thank you. Thank you to all of you, because all of what we're going to talk about today, all of the research that we do is only possible because of families like Jack's family and supporters like yourself. So thank you very much, and I hope you have a great evening. At Boston Children's Hospital, we have a research program for hereditary spastic paraplegia. In our program, we support families with hereditary spastic paraplegia from around the United States, around North America, and around the world. Our research program integrates clinical research and care. We meet with families from around the world. We gather natural history data. We identify biomarkers, and we work on developing therapies. Our program is based in Boston at Boston Children's Hospital and Harvard Medical School.
And I would like to walk you through how we meet our patients for research. So with the first concerns and symptoms, patients show up in our movement disorders clinic. We then have the option to enroll them in the HSP Genomic Sequencing Initiative, where we provide advanced genomic testing. And the goal is to discover genes for unsolved movement disorders and provide a diagnosis for those patients. With the diagnosis, we can provide clinical management and enroll them in the natural history study of childhood onset HSP, where we describe the natural history study of early onset HSP, work on biomarkers, and in the next step, we hope to expand this to our now established multi-center study, the Spastic Paraplegia Centers of Excellence Research Network. And this is really a combination of clinical care and research. And the overall goal is, of course, to improve therapy for our patients.
Our research program is embedded in a pipeline that is looking to take discoveries all the way from a preclinical stage to a clinical stage in clinical trial. Drug development for rare disease is complicated. It requires a lot of infrastructure, it requires collaboration, and it requires a team. Where we hope to make a difference is in providing both clinical trial readiness as well as testing novel therapeutics in the lab, in disease models, in cells, in animals, and then together with partners take these discoveries all the way to a clinical trial. We're very aware that this is a long journey and requires a lot of resources. So with your contributions tonight, you're making all of this possible.
And I want to emphasize that a major partner in this are our patients and their families and their communities, including Jack and his family. In fact, Jack was our first patient in our multi-center study to gather the natural history of hereditary spastic paraplegia. What families do in these studies is help us systematically characterize SPG4 by sharing their experiences through yearly study visits. Families in our genetic testing study help us explore genetic features that potentially tell us more about the disease and what we may consider for a potential gene therapy. And I also want to say that outside of research, patient families and their communities have strengthened the research collaboration that we have, and by encouraging the exchange of research, raising awareness, and of course, raising funds. And this is all to push forward the knowledge around SPG4 so that we can help these kids. Thank you.
So with this, a big thank you from our team, and we hope you have a great evening. Thank you.
Thank you.
Hi, I'm June, and I'm Jack's friend.
Hi, I come in. I Jack friend. Hi, I'm Sam. I'm Jack's friend. My name is Nathan, and I'm friends with Jack. Hi, Jack. I miss. Hi, my name is Tanya, and I am Jack's friend.
Hi, I'm Lily. I am Jack's friend.
Hi, my name is Maria, and I am Jack's friend. Please help Jack and his friends who has SPG4.
Hi, my name is Rio, and I'm Jack's friend.
Hi, I'm Ellis, and I'm Jack's friend. And I have SPG4 too. Thanks for helping us.
My name is Jack. Thank you for helping me and my friends.